Okay, let's start. Good morning, afternoon, evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa. 안녕하세요. Hello all. Welcome and thank you, SynBYSS people, for joining us, the 56th seminar, and I call it a BioMADE Day. I just finished my 2 weeks of seminar trips until now, and will resume 3 weeks of international trips to give 6 more invited talks, including a keynote talk at a conference. I'll meet you from Asia, probably at midnight or 2 a.m. Still, I am so excited to meet many people in Asia in person. Okay, it is my tremendous honor to introduce very briefly, but I'm not sure I can briefly introduce him, our pioneer speaker, Dr. Clem Fortman. Dr. Fortman is the Director of Program Management for BioMADE, a US DoD supported organization to enable translation and commercialization of synthetic biology through biomanufacturing. I have known him longer than 15 years. I met him at multiple meetings when he and I were the members of NSF SynBYSS, the first synthetic biology center in the world. As you know, BioMADE was created by people in EBRC, Engineering Biology Research Consortium, where I serve as a council member. The CEO of BioMADE is Doug, and he was our EBRC director when SynBYSS became EBRC, by expanding its membership to more people. Because of this close connection and collaboration between EBRC and BioMADE, I am tremendously happy to have Clem today as a pioneer speaker. And Clem is passionate about those biotechnology translation. At the same time, Clem is care about people, especially young people. So it's my great pleasure to have him today. So Clem, the virtual podium is all yours now, and thanks so much for your time today and contribution to the society through many other organizations, including BioMADE and also SynBYSS and EBRC. Thank you.
All right, thanks so much, Professor Moon. I am truly honored to be here among you today. I will say what I would like to say philosophically. As usual, J.P. Kiesling has already said more eloquently than I ever could. So I will, if you want to know about my philosophy, go and watch his intro. I will instead take this opportunity to tell you about my somewhat non-traditional career path, and hope to convince you that there is a place for you in this community. It doesn't matter who you are. If you're interested in being here, we want you here. You are part of this community. I don't think of synthetic biology so much of a field of study as it is truly a community, thanks to organizations like SynBYSS and EBRC. We work together very well, multidisciplinary people, even artists coming in to contribute what they have. So I will start, and I will hammer on 2 strategic points that I think are important for moving forward in your career. One is to take advantage of the opportunities that you're presented as you move up from undergrad to grad school to postdoc, if that is the trajectory you take. There will be opportunities for you to work with people, to mentor people. And I would suggest that you take advantage of them. The second point is to give back. So the mentoring part along the way, once you start learning things, start teaching things. It helps you learn better and it helps you support the field. So my career trajectory is probably not the same as most of you. I got kicked out of high school when I was 17, and I went into the Army. I went in as a radioman and was provided some opportunities to learn chemical and biological warfare. So my first biology class outside of high school was to learn about bioweapons and how to protect against them. I also went to night school and got my EMT license and cross-trained as a medic. So I started learning the fundamentals of medicine. I got out of the service and did this sort of usual post-service enlisted man kind of work. Everything from roofing to bouncing to ultimately I managed a bookstore for a long time until my wife convinced me to go to college. So I started out in the junior college system and worked my way through taking courses at a community college, and then transferred in working full time. I think I worked 6 days a week for the first 5 years of my undergrad. Started doing directed research in synthetic chemistry for Professor Wayland Noland at the University of Minnesota, who is, I believe, 95 and still mentoring students and turning out papers. So he continues to give back after all these years, and was quite an inspiration for me. So I had the opportunity to work for him. And then I did that for a few semesters, got reasonably good at organic chemistry, started TAing, started giving back mentoring. I went into college thinking I would go to medical school. I was interested in microbiology. I looked for opportunities in microbiology. I wanted to work on tuberculosis because it's the most important bacterial pathogen on the planet. Nobody at Minnesota at that time worked on it, so I got steered towards streptomyces because of where it sits on a phylogenetic tree. Sort of ridiculous, but there it is. Started studying secondary metabolite biosynthesis with David Sherman. Got reasonably good at that. I had some aptitude. I really liked using microbes to do chemistry. I went with him when he moved from Minnesota to Michigan, and ultimately did my grad work with him, and then moved on and did a postdoc with Jay Kiesling at UC Berkeley. There I was afforded a lot of latitude to do things. I ended up doing a lot of work in biofuels, but I was also fortunate enough to be able to develop an educational outreach program that's called Clem, because I can talk a lot of smack. And myself and James Carruthers developed the original nugget of that. So this was us giving back to those around us and to the high school kids that are coming behind us. That in itself became its own community. We had people volunteering, grad students, postdocs, technicians, everyone in the community recognizing the power of teaching these high school kids. So that was emotionally the most gratifying thing I've done thus far in my professional career. On the scientific side, I developed a lot of patents and technologies around polyketide engineering. Jay and I and a few friends launched Lygos, one of the many startups in Jay's portfolio. And I was president and CEO of Lygos for a while. I found that I'm not as financially motivated as I thought I was, and decided to go back to a more mission-focused job, and I went to work in the Pentagon, where I worked on chemical and biological defense. So circling back, service to the nation, using both my technical expertise in synthetic biology and my knowledge from the previous life in chemical and biological weapons. I worked in that area for 2 years and then was given latitude to help do community building around the synthetic biology stakeholders in the Department of Defense. So getting that community started to coalesce together because there were a lot of independent efforts. For those of you that don't know, the Department of Defense has funded a lot of work in synthetic biology for a long time. It's been quite supportive of this field. They recognize that as early adopters of technology, they'll be able to take real advantage of things that are emerging. So bringing them together a little more, I started a DoD-wide newsletter that talked about synthetic biology that still is issued every 2 weeks, even though I'm long gone from there. So I brought the DoD internal community together. I tried to bring them closer to the external synthetic biology community. I got them involved with the EBRC, and then I switched sides and went to work for Doug at the EBRC, doing essentially the same thing from the other side, trying to teach that community about biosecurity. And then the DoD went and issued the call for BioMADE. I helped put together the proposal there, and now am spending my time helping move relevant projects, not just DoD, but projects that will help the whole community develop technologies to move the non-medical biotechnology, synthetic biology projects off the bench and into the marketplace. And I'm still looking for ways to give back. So thank you, Professor Moon, for this opportunity. BioMADE does work in the education space, so some opportunities for me to give back there as well. But I am very grateful every day. I get up and am able to interact with folks like you and help move this community in whatever small way I can. And I will encourage you. I mean, if you see Jay's talk, he says, find your passion. I'm still kind of looking for my passion, but until then, I'm having a lot of fun and recognizing that there is room for everybody here. If you don't like working at the bench and you want to go to law school, there's a lot of patent work to be done. If you want to go to business school, you can help this field develop in very substantial ways. When people make money, more money comes into the field, better research gets done, more things happen, more bioproducts are going to help save the planet. It's all, I mean, it all comes back together, but only through truly broad community efforts. So find your spot, find your happy place, and reach out to me if you want to talk more. And with that, I will cede the floor back to Professor Moon. Thank you so much.
Thank you so much. You know what? I agree with you every single point you made. And of course, I mean, you could contribute to the society by, doing science, but you could also do something else like the business, even patent law, I mean, multiple different ways. And I believe you are true demonstration, people could, demonstrate, I mean, also contribute to the society because you already been multiple different places. I mean, DoD, and now biomanufacturing, organization to help people to come up with new idea and biomanufacturing and all kind of different things. And then I was impressed by your trajectory. I didn't know actually until today you go through very unusual or rare kind of trajectory you had. But at the same time, I realized you and I have many common things I didn't know, I mean, until today. For example, I already knew, I mean, Jay, is kind of your advisor, but Jay is my academic grandpa. So that means you are my academic uncle in a sense. And also, you mentioned Dave Sherman. I didn't know about that one, but when I was the master's student, 1998, my mentor was actually Dave Sherman's, advisee, Dr. Ung-Soo Kim. And Dr. Ung-Soo Kim teach everything about SynBYSS at the time. So, I didn't know, that connection also there. So I would say, human is all connected somehow, one way or the other. I was so surprised, those direct bond. Also, you mentioned about DOD supporting a lot of synthetic biologists. In fact, last 10 years, out of my 21 grant, actually 6 grant is coming from DOD, including 2 DARPA, 3 ONR, and 1 Air Force grant. So I'm very grateful their support. So thank you so much. This is absolutely brilliant, talk. And then, I believe many people kind of get some inspiration out of your talk. Thank you so much.
Yeah, it's my pleasure. Thank you.
Yeah, thank you. Okay, so now the main speaker of today with a longer introduction. Also, I found just now, she is my academic nephew, right? So Dr. Carolyn Mills is a postdoctoral fellow at Northwestern University in Professor Daniel Turman-Eric's lab, and she's actually my academic sister through Chris Voigt, and where Dr. Mills applies protein engineering principles to self-assembling protein system. She completed her bachelor degree in chemical engineering in 2013 at the University of California, Santa Barbara, UCSB, and received her PhD in chemical engineering at the MIT. Of course, she's my alum. So I also went to same department, same university. As an NSF graduate research fellow, Dr. Mills completed her PhD in Professor Bradley Olson's lab. I think his lab is next to my old lab, but now my advisor moved to another building, where she investigated self-assembly and high-throughput processing of fusion protein materials. Her current work focused on protein nanoreactors called bacterial microcompartments and how these nanoreactors can be re-engineered for metabolic engineering applications, as well as how they might serve as targets for future antibiotic development. She's truly a rising star with huge potential. In fact, I met her this year multiple times and place, including a conference in Cancun, where we discussed science and life together, and also others, the topics. So Carolyn, thank you so much for your time today, and please take it away. It's all yours now. Thank you.
Thank you so much. I just want to thank Clem for his contribution to. I didn't know you were also, I also went to community college before going to my university, and I'm always telling people what a great choice that was for me. So I'm really, that's such a cool thing that managed to overlap. Let me get my screen shared here. If it will work. Can everyone see what's going on? Actually, 1 second, sorry. Oh. Let's see. Oh. There we go.
Yeah.
Can everyone see? Okay,
Yeah.
now it's looking good. Okay.
Wonderful.
Good morning, and thank you to TASOC for organizing this seminar series. I've been attending since it started, depending on where I'm at and if I am available at the time of the speakers, but it's been really fun to just hear about what all the young people who are going to be kind of doing the next stage of synthetic biology are going to do. So I've had a great time coming to this and seeing the people who, you know, presented last year now getting awards and faculty positions. Very cool. Great eye for people. Today I am going to talk to you a little bit about our work on bacterial microcompartments. As TASOC mentioned, I am a postdoctoral fellow in Professor Danielle Tolman-Ersich's lab at Northwestern University. And get started. So to start, I would like to point out that spatial organization is really important. And this is true in our cities, our homes, and our offices, where spatial organization is kind of the magic sauce that keeps things running smoothly. But it's not just true that spatial organization matters in these facets of our lives, it also matters a lot in biology, and it's really essential in nature. From the subcellular organization that we see in cells to the organization of cells into tissues that perform different functions in our body, like the brain, to the spatial organization that multiple organisms coordinate with each other to form these ecological communities, it matters a lot. But in our lab, we actually work with bacteria. And when you think about bacteria, you might think that these are things that totally lack spatial organization. They are messy. But in our lab, we believe that fully harnessing the potential of bacteria as tiny reactors, producers of chemicals, requires us to be able to spatially organize processes within them and to be able to engineer this property. And we have some reasons why we believe this is important. So we believe bacteria are great chassis for performing these kind of metabolic engineering applications where you can reprogram these bacteria that grow really quickly by putting enzymes in them. And these enzymes can perform a set of reactions that can make things like value-added chemicals, pharmaceuticals, and biofuels. And this is possibly even more exciting when you think about the fact that there's a ton of enzymes out there and there's more and more functions that are being accessed. I don't know that I would say every day, but probably every day using stuff like directed evolution. But having these enzymes alone and being able to perform these reactions is not quite sufficient in our mind. Because while these bacteria they grow really fast and are highly genetically tractable, if we can't really organize these processes, we can run into several problems. And I'm kind of summarizing a couple of here. So you can imagine that in your process, if you're trying to get from, say, A to C, you may, on the way to making C, make something that's toxic to the cell, and that would kill your host. That's not good. You can no longer make a bunch of bacteria that can make your chemical of interest. You may also run into issues of competing for resources, either in the form of your intermediate goes into some sort of garbage product that's not what you want, or you can have competition for things like cofactors that, again, decrease your product yield. If we are instead able to encapsulate these processes or sequester them away from the rest of the cell, we could obviate kind of all of these challenges. Toxic intermediates would kind of be sequestered away from the rest of the cell, not killing it. And alternatively, these processes would also be sequestered away from the cell, preventing things like side reactions or competition for cofactors. So with that in mind, one of the ultimate goals in our lab in this metabolic engineering space is to be able to control the spatial organization towards enabling bioproduction in our cells, in these bacteria that are highly genetically tractable and grow really fast. But to achieve that, we first kind of need to understand how to program this organization. And this raises a question of what is the best way to do this? And it turns out that we were not the first people to have this idea. In fact, we got scooped by Nature many years ago. And however, it's very cool, these things called bacterial microcompartments exist. And bacterial microcompartments are these subcellular organelles that already exist in bacteria. And I'm showing you some micrographs of them here. You can see there are these like polyhedral things in the stem cell section. If they're purified out, you can see them little better. They're these irregular polyhedra that have this characteristic shape. And more specifically, they are these organelles made up of a protein shell that encases an enzymatic core. And in this enzymatic core tend to be processes that take a given carbon source and convert it into energy for the cell, where the carbon source that goes into these bacterial microcompartments tends to be some sort of niche weird carbon source that has to pass through some toxic intermediate on its way to becoming energy. And so by sequestering away the processes that produce and subsequently consume the toxic intermediate, the cell can process these carbon sources without suffering these kind of bad side effects of the toxic intermediate buildup. And for me personally, but also I think as a protein engineering lab, what I think is very cool about these microcompartment systems and what makes them, I think, really great targets for engineering is that they're actually completely genetically encoded because they're made up entirely of proteins. So theoretically, using this whole suite of tools that we now have as synthetic biologists for genetic manipulation, we could make whatever changes we wanted to this bacterial microcompartment in a relatively straightforward manner. So this all sounds really cool, but there is probably always a catch. So what is that catch? The catch is that bacterial microcompartments are complicated. I'm showing you here the operon, the PDU operon for the model bacterial microcompartment system that we work with in our lab, the 1,2-propanediol utilization, or PDU operon. And as you can see, it's made up of a lot of different proteins. There's about something like 20 genes on here, and that includes enzymes that perform things like the main pathway, as well as cofactor recycling, as well as all the proteins that make up this shell that encases these enzymes. And the fact that these are complicated and there's a lot going on means that reprogramming them can be a little bit difficult because we don't know which of these are most essential for forming the bacterial microcompartment that's so essential for spatial organization. And we don't know if we make changes in one place, are we going to affect some other process. So the goal of this work is a little more fundamental then, and it's been to identify an assembly mechanism for this microcompartment, identify the protein-protein interactions involved in these assembly stages so that we can engineer it without disruption. And finally, to develop high throughput tools for screening for this assembly, which also has implications, as Tessa kind of mentioned in my introduction, in antibiotic development, because these microcompartments tend to be associated with pathogenesis in some bacteria. So to start, we want to find an assembly mechanism. And at first glance, that kind of leaves us here with some knowledge about the protein units that make up the compartment and an idea of its final composition and shape. But the question then becomes, which of these are essential and what do they do? And where do we start? So this actually started a little bit before I joined the lab with my lab mate, Nolan Kennedy, and he decided to start by looking at these two proteins, PDU-A and PDU-J. And he had a good reason for it. It wasn't just random. These two proteins contain these hallmark bacterial microcompartment or BMC domains that are, as I said, a hallmark of bacterial microcompartments. They're the domains that form these six-sided tiles that form the microcompartment shell. And these two in particular are the most abundant ones in the PDU microcompartment. So he figured they're probably special in some way, or at least essential. And almost immediately, what he found is that these two proteins, PDU-A and PDU-J, have this remarkable self-assembly property where they self-assemble into these super long tubes that hopefully you can see in these transmission electron micrographs here when they're on their own. So when you overexpress these two proteins in the absence of anything else in the cell, they form these super long tubes that self-assemble. And what Nolan thought when looking at this was, okay, maybe it's the self-assembly property that initiates microcompartment assembly. And it turns out that he was correct in this regard, but before I get into that, I just want to go over these kind of a couple micrographs that we're going to be seeing a lot in this presentation, so I want to cover what you're looking at here. So in the micrograph on the left, we have a phase image right here that tells us where our salmonella cells are. In the middle, we have a GFP micrograph, and that tells us where the compartments are within our cell because we've put a little tag on them, a GFP marker, that localizes to the interior of the compartment. And a well-formed compartment is characterized by punctate fluorescence throughout the cell, as you can see in the spots of the case. We also look at these a little bit more in vitro, where we purify them out from the rest of the cell and then image them with transmission electron microscopy. And a good compartment, as hopefully you can see here, is characterized by these kind of well-defined polyhedral edges in these purified compartments. Going back to our hypothesis that the self-assembly property drives microcompartment formation, we can see that if you knock out, that's what this delta is here, knocking out each of these individual proteins, that spoiler alert, that you actually still get well-formed compartments as characterized by the fact that this GFP micrograph and these purified TEM images both pretty much look like our wild type case. And this is why this is a little bit tricky, because on their own, getting rid of one of them doesn't ablate compartment assembly because the other one can kind of make up for it because they both self-assemble. And it wasn't until we got rid of both of these self-assembling proteins that we saw kind of an ablation of compartment assembly. And you can see that here in the GFP micrograph in the form of these, what we call polar bodies. So instead of punctate fluorescence distributed throughout the cell, we have our cargo aggregating and getting pushed to the pole, which are these kind of GFP blobs that are existing at the pole of the cell. In our purified compartment system, all we got were little kind of fluffy blobs that are not characteristic of our well-formed compartments. So this gave us a starting point for our assembly process and told us that, then suggested that PDUA and PDUJ self-assembly initiate the formation of our PDU microcompartment system and likely initiate the assembly of other compartment systems as well. But that is just kind of one component, the first component. We kind of have 18 to 20 other things that need to get incorporated into the microcompartment. And so the question becomes, how do those other things get in? And how do we go from these hexamer self-assembled structures into a well-formed compartment? I'm going to start by talking about these other hexamers as well as the cargo and asking, how does this enzymatic cargo get incorporated into our compartment? So I worked on this project again with my lab mate, Nolan Kennedy, shown here. But when we first started to look at this and ask the question of how does cargo get incorporated into compartments, we went to the literature and we saw that there was a lot of conflicting evidence. Almost every single shell protein in here was implicated in recruiting a different type of cargo, as hopefully you can see in these references I'm showing in this figure here. All of these different kind of ovals in the middle are different enzymes that are encapsulated in the microcompartment. And so we thought this is really complicated or there's something that we're not seeing here. And based on the literature and doing some kind of closer looks at data, we decided to go with what we thought was the most promising and probe the hypothesis that it was one protein and one protein alone, PDUB, that was responsible for actually bringing all of the cargo into the microcompartment. And to test this hypothesis, we built this suite of fluorescent reporters that pretty much report on every different enzymatic cargo that can get into the microcompartment, and I'm showing here. And this would tell us if it was just one thing, like one specific cargo component that's localized to the compartment with this PDUB protein, or if it's all of them. So we're going back to our microscopy assay here, and what you can see in our positive control or wild type case is that, as expected, all of our different types of cargo exhibit GFP tag cargo, localized to compartments as you can see in this, the punctate fluorescence we see in these fluorescence micrographs here, showing us that indeed all of these things are associated with compartments and our fluorescent reporters are behaving as expected. So that's our positive control. These are all getting in normally. If we knock out PDUA and PDUJ, that's our second row here, compartments can't form, as I showed you in the previous slide. And again, with all of our different reporters, we're getting this kind of aggregated cargo that localizes to the pole of the cell, as we kind of expect. So then comes our experimental case where we're getting rid of that PDUB shell protein. And what we found is that all of our different shell protein reporters, or cargo protein reporters, now are localized to the pole of the cell. They're no longer punctate. This could be the result of one of two things I'd like to point out. So it could be that compartments can't form anymore and all of our cargo is getting pushed to the pole, as is the case in our PDUA, PDUJ, broken compartment control. But it's also possible that the cargo can't get into compartments anymore, and because it can't localize to the interior of the compartment, it's aggregating and again getting pushed to the pole, as I'm kind of illustrating with this diagram here. To distinguish between these two possibilities, we turn to a second fluorescent reporter, our friend M. Cherry, And we decided to tag the shell of our microcompartment so that we could see where our shells and our cores of our microcompartments were going, and if they were going to the same place. So in the case of our wild type strain where compartments are forming well, you can see that the shell protein reporter, PDU-AM cherry, exhibits punctate fluorescence as we would expect. There's puncta associated with shell proteins distributed throughout the cell. And that a cargo protein, SSP, GFP, is also punctate in the cell. And importantly, that when we merge these two images together, that both the shell and the core are localizing to the same place, suggesting that cargo is successfully getting localized to the interior of these compartments. We then compare this to our PDU-B knockout strain. You see that while we still have punctate fluorescence in our shell protein reporter, suggesting that our microcompartments are still forming shells, our cargo protein reporter is now localized to the pole of the cell. And when we merge these two images, nothing is localized in the same space. So our core is completely decoupled from our microcompartment cell. And to confirm that the puncta we're seeing in this particular micrograph were in fact compartments, we performed transmission electron microscopy on purified components. And we found that indeed in this PDU-B knockout strain that we were still getting these characteristic polyhedra forming, confirming that PDU-B was not essential for shell formation, but instead was essential for bringing cargo into the microcompartment shell and kind of linking those two processes together. This established our second step of microcompartment assembly. PDU-B links the self-assembling proteins PDU-A and PDU-J to this entire enzymatic core. We kind of have a final question in the puzzle, which is how do we go from those sheets wrapping around cargo into this closed polyhedral shape? What is the final step in our assembly mechanism? This is where my favorite protein in the PDU operon comes in, PDU-N. And I would like to introduce PDU-N by revisiting our microcompartment operon. So of the shell proteins in this operon, shown here in blue and green, all of them except for one are made up of these bacterial microcompartment or BMC domains that form 6-sided hexagonal tiles. But there's also one gene, PDU-N, that forms instead a bacterial microcompartment vertex or BMV domain protein. And these domains form 5-sided pentamers that are thought to cap the vertices of bacterial microcompartment systems. And for this reason, we identified PDU-N as a likely candidate for mediating this final vertex capping process and making our PDU microcompartment into a closed shell. So with this knowledge in hand, we set out to characterize the role of this PDU-N protein in the assembly of these microcompartments and hopefully get at that final piece of our assembly puzzle. So I'm bringing back our favorite microscopy assay here. And what you can see here now is that instead of observing polar bodies or puncta in our GFP micrograph, we instead see these striking elongated structures when we knock out PDU-N. So compared to the punctate fluorescence that we see, in our wild type compartments, we now have these long, what may be tube-like structures that seem to be traversing the entire cell. And to confirm that this was the case, we used thin cell section TEM to visualize where structures were within the cell. And it's a little bit hard to see here because the contrast isn't perfect, but in the wild type case, we see compartments that are distributed throughout the cell. And in the PDU-N knockout case, we instead see these tubes that are aligned on this long axis of the cell and appear to be kind of bundling together along that axis, which hopefully you can see in these little plots there. And we confirmed this using by performing TEM on purified structures as well. And in this way, we were able to confirm that these were tube-like structures that were about 50 nanometers on average in diameter, suggesting that we're getting these compartment structures in the wild type, but these weird tube structures in this PDU-N knockout case. And what's interesting about these tube-like structures, to me at least, which you can hopefully kind of see with this GFP micrograph, but also by SDS page, which I'm not showing here, is that these tube structures contain most, if not all, of the same components except for PDU-N as regular compartments do, suggesting that instead of being like some weird offshoot structure, they're actually mostly just compartments that are long. and that this vertex capping process that PD-UN mediates is actually the final step in our compartment assembly process. And with our previous work that I've kind of talked about a little bit today, this brings us to our proposed assembly mechanism for these PDU microcompartments. It starts with this hexamer self-assembly that initiates by forming these hexameric sheets. PDUB then comes in and starts to wrap these sheets around cargo, imposing some shape constraints on this process. And finally, PDUN comes in and facilitates vertex capping of these compartments, facilitating their formation into these closed compartments. So this concludes the first part of my talk about our investigation into the PDU microcompartment assembly mechanism. But I also promised something about high throughput techniques and how we want to be able to screen for whether each of these assembly processes is happening correctly as we start to engineer and make changes to our compartment. So while I don't have time to talk today about each of the high throughput assays we've been developing for each of these stages, where I promise that we have been developing them for each of the different stages of this assembly process. I want to give you a flavor of what we've been working on by specifically going over the screen I've been developing for this third stage of microcompartment assembly, this vertex capping stage. And to get into that, I'm going to return to our micrographs here and point out something that you've probably already noticed, in that in these phase micrographs in this PD-UN knockout strain, you may notice that there's a difference in cell phenotypes between our wild type cells and this PD-UN knockout that forms these elongated tube structures. And I'm recapping that here with an illustration that cells that express compartments where vertex capping occurs correctly, that's our wild type, result in these unlinked cells that are the length of normal salmonella. And by comparison, when we knock out this PD-UN protein, we get these tube structures that create a linked cell phenotype. And using microscopy and some image analysis, we confirmed this to be the case, and that we found that there was a statistically significant difference in cell length between cells exhibiting this proper vertex capping and those that couldn't. That's that PD-UN knockout there. So this is all well and good, but how does this translate into a high throughput assay? Well, you can imagine that if you express your PDU operon and proper vertex capping occurs, regular compartments will form and cells are not going to be elongated. On the other hand, if our vertex capping does not occur correctly, either due to a knockout of the PD-UN protein or say a mutation or addition of a drug that is not compatible with PD-UN incorporation into microcompartments, we expect these extended tube structures to form, resulting in an elongated cell phenotype. And as you can hopefully see in these images, this phenotype is easily distinguishable by microscopy. So to illustrate the utility of this and see whether it was a robust assay, we applied it to investigate two residues in our PD-UN protein. One that we expected to be highly immutable, that's this G52 residue here. It sits kind of nestled in this interface that is expected to be responsible for PD-UN incorporation into the shell. And this other residue shown here in yellow is expected in contrast to be highly mutable because it just kind of sits at the top of that interface poking out, and mutating it to most other things shouldn't really matter, was our thought. So when we mutated this G52 residue, that's the one we expect to be immutable, what we see by microscopy is that indeed mutating it disrupts this interaction in the incorporation of PD-UN, which is indicated by this linked cell phenotype. And we confirm this using our other kind of TEM techniques, showing that we have these elongated structures forming when we make this mutation to PD-UN. In contrast, mutation of this threonine residue that kind of sits on the top, we expect it to be mutable, results in unlinked cells containing punctate GFP fluorescence indicative of proper compartment formation. And we confirm that to be the case, again, using transmission electron microscopy. So thus far, I've shown two single point mutations that we analyzed with this whole suite of techniques to confirm that what we expected was correct, illustrating hopefully to you the utility of this screen, but I promised that this was a little more high throughput, so I'm going to show you now data measuring this linked cell phenotype for all possible point mutations at these two positions. So here I'm showing you a summary of the screen performed on these 2-point mutant libraries, where the x-axis indicates the point mutation made to our PD-UN protein, and the y-axis indicates the percentage of linked cells observed by microscopy. which is essentially a measure of that elongated cell phenotype I described. When PDUN can incorporate, long structures form, resulting in highly linked cells. When vertex capping happens properly, compartments form and cells don't form elongated chains. And this comprehensive point mutant data shown here for these two residues shows that indeed G52 is highly mutable, illustrated by the high percentage of linked cells across all point mutants. Where there's this one kind of slightly interesting partial exception, this G52N mutant, which exhibits a statistically significant lower percentage of overall linked cells. And we hypothesize that this asparagine mutation might be tolerated better due to its ability to form these backbone-like hydrogen bonds that are characteristic of glycine. But again, that's just a hypothesis. It was just kind of interesting to see that we could get something that was a little bit more quantitative than just totally linked or totally unlinked by this assay. Conversely, if you look at the point mutants in the T88 residue, we see that this position tolerates all mutations relatively well, evidenced by the fact that we see very few point mutations of any linked cells at all. But kind of taking a step further back, what this data really shows us is that we have this rapid, robust, and semi-quantitative assay for investigating disruptions to this particular stage of compartment assembly. And importantly, this is going to enable future studies with larger point mutant libraries that can precisely interrogate the interactions responsible for this vertex capping stage of this complex compartment assembly process. Towards our goal of engineering multi-protein assemblies, this data is going to provide, in conjunction with these kind of larger libraries, crucial groundwork for building design rules around these complex assemblies and allow us to rapidly screen for mutations that interrupt this particular stage of assembly. And importantly, it will also allow, with the other tools that I wasn't able to tell you about today, we could theoretically probe all of these stages of assembly in a similar manner using microscopy-based techniques. We're also looking to extend this to work with things like fluorescence-activated cell sorting so that we can screen libraries in higher throughput. And with that, I would like to thank our funding sources, the Department of Energy and the Army Research Office, as well as the entire Tolman Ersic Lab, calling specific attention to both Nolan Kennedy and Alexander Jackson, who both provided data for the talk that I gave today. And while I didn't have time today to talk about any of the work we've done with our collaborators, I want to call out all the collaborators who did work on the papers that came out of this work, specifically the Olvera de la Cruz lab, the Mangan lab, and the Jewett lab, for their help in some other interesting stuff we've been doing with these compartment systems. And with that, I would be happy to take any questions.
Okay, wonderful, wonderful talk. So that's absolutely great. I wanted to ask Clem whether you have any comment or question for her, especially challenging one.
I'll accept easy ones too.
I do not. I'm too far from the bench to dig in hard on these. I'm glad to see the microbial compartments being engineered successfully. I feel like I was around for some very early efforts and saw the promise, and am glad that the promise is bearing fruit, because this is, we've seen many processes that hit toxic intermediates, and being able to compartmentalize the biosynthesis of stuff in the future is going to be, this is going to be super powerful, especially for things like pharmaceuticals, where you really can get some hardcore toxicity of intermediates and even final products. So thanks for pushing this forward.
Okay, so I wanted to ask some question. So I see the great potential using your approach in this specific bacteria, and I just wondering whether the same approach or or similar would be generally applicable to other. In that way, the approach and could be generalizable. Could you kind of comment on this point?
Yeah, and I think that's a great question that we're, all of this is still like like kind of laying the groundwork of understanding these. And I think there's probably two ways you could go about it. One way is obviously just trying to take something that we already understand very well and port it over into a new bacteria, something that may be a better host for your process of interest than Salmonella. But another thing that's really interesting about these compartment systems is that I wouldn't say they're ubiquitous, but they exist in a lot of bacteria. Whether they're functional or not, or we have the tools to study them, or like the tools to manipulate them in the same way we have these, is a little bit further off. But I think that's another option and you can kind of consider doing both, either porting this entire thing over or trying to re-engineer ones that exist in your target bacteria of interest should you have one. And this is something that we're kind of exploring. It's one direction that the field is going, kind of figuring out what other compartment systems exist and then starting to characterize them so that now we can make manipulations. So yeah, I do think that there's progress being made in both of those areas, and making a decision on which will be the best one may depend on things like what organism you're working with. If you're working in a bacteria that has totally different codon usage, for example, or has a lot of different regulation, you may want to use the native system, because you could totally, if they have a native compartment, you may want to re-engineer that. But yeah, I would say because they exist in so many different types of bacteria, I'm very tempted to say that they should still form in most bacteria, and that is, it's something that's interesting to us as well. You could also think about taking these compartment systems because they can help give this competitive growth advantage, or that's the thought, and putting them into a probiotic, giving them that ability to grow on these carbon sources that pathogenic bacteria can currently grow on. So that's another place where we might want to like port it in and use something for something else. But yeah, it's a good question, but I think there is actually a lot of ways to start thinking about doing it. And choosing that strategically is also important.
That's wonderful. So as you mentioned, there will be two ways. That's also I was thinking about, but at the same time, I just want to hear from you what would be, challenges or, more, kind of, advantage kind of using each method, kind of, for example, using native system versus, your system you already developed, basically transport that into the new organism.
Yeah.
You mentioned some of that, including codon, optimization and so on. But I mean, beyond that, I mean, what kind of challenges, each has, and what kind of, advantage each has? Could you kind of comment on that part as well?
Yeah, for sure. Happy to get these questions because we think about it a lot. So I think the advantage of using one that is well characterized, so kind of taking a model one and moving it over, is that we have we have a very deep understanding of this assembly process. We know what matters, and you would have to we have a lot more tools and we can do it more quickly now, but characterizing a new system requires kind of redoing 15 years of work in a couple of years, Again, we have a lot more knowledge to build off of, but you would still kind of have to confirm all of these steps. So I'd say that's the advantage of bringing over a known system, is that you have a very good idea of how to engineer it. I think the challenge of bringing over, porting over a system is things like regulation and control, and ensuring, as I said before, codon optimization is one thing, but you also would need to find a way to, induce the formation of these things, and you have to consider the promoters and all that kind of stuff that's going to be existing in this new microbe. And because this is such a complex process, those things can be kind of sensitive. It's very sensitive to some of these changes in regulation. So that's my take on the things that are challenging at each stage.
Yeah.
Does that answer your question, or were you?
Yeah, that's a completely refactoring, question. I was working on refactoring, more than 10 years ago in Chris Boyd's lab.
Oh, you're part of that. Okay.
Yeah.
Yes.
So that's why also I'm thinking about, just using native system, but we don't have, any understanding about native system. Also, some bacteria even not probably having those kind of compartments. That's why I'm asking that question.
Yeah. Yeah.
Okay, so Clem, do you have any other thing to ask or comment? Or if not, I may close because I didn't see any question from general audience.
Actually, I just have one it will be pure speculation on Carolyn's part, but so you say it would take redoing 15 years worth to characterize, but you've got a good base here. Like if you found this in a bug that was promising for other attributes, carbon utilization or solvent tolerance or something that helps a larger process, what do you think is a realistic timeline for getting to this point with a with a novel organism, like studying the native system and building up to at least where you are now?
Yeah. With infinite people working on it, if we were to say that. If we have
Frame it in FTEs like a proposal.
I was thinking about how much I want to say. We actually do. We are the DOE funded proposals. I think it is public knowledge. We are working on this with Lonzatech on their Clostridium system, trying to re-manipulate this so I can actually directly attest to all the like different things that occur in this case. I think, to get to this point where we're not putting new pathways in and we have the same level of understanding, probably, a couple years, I would say. And because we do have so many tools and we have so much knowledge, I think if it was someone who is not like a compartment expert, it would probably take a little bit longer to kind of get acquainted with the subtleties of it. But, I'd say probably 2 to 3 years to get that off the ground, to understand how to manipulate it, and then another couple to, if we wanted to start putting new pathways in, because that's still something that we're working to kind of completely understand in the native system, is this reprogramming part. But yeah, I do think that the tools and knowledge that we've built up so far really accelerate it, and this familiarity we have, and yeah, that would be the hopeful estimate.
Mm-hmm.
Yeah, that's great. I mean, I just want to point out, it's like another useful tool for engineering biology. There was a point when characterizing and manipulating a newly discovered plasmid was a 2-year endeavor. So, just put it in context. It's rolling forward, and if it shows a lot of utility and adoption, it can blow up. I really like to see these sort of basic research technology focused things maturing. It's exciting. Thank you.
Yeah, thanks for the question. And I think that's a great point. And that's the point we're hoping to get to eventually in these systems. It would be really cool.
Yeah, sure. I mean, Carolyn, once you start your position, ask $30 million to him just to start your project, or $5 million. Why not? He has money. All right, so I will close. Let me double check one more time. Okay, so I'll close. Okay, so thank all for joining and staying today. We'll meet again next week on October 27th. Oh my God, already end of the October. Thursday, the same time, the same Zoom link. We'll have Professor Barack Cohen at Washington University Medical School. Finally, the first sort of kind of the professor from WashU, and Dr. Christian Euler at University of Toronto. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us, today's speakers. I'll promote you to panelists who can speak and show your handsome and pretty face. And thanks. I'm stop recording. Just give me 1 second. Just stop recording.